Life sciences involves the study of microorganisms, plants and animals, and research in this area is of huge importance to society. We need sustainable food sources, and we would like to be as well armed as possible in the event of something like a pandemic, or if ourselves or a loved one became seriously ill. My lab studies the molecular workings of human cells. Our cells are full of proteins, and many of these are enzymes whose aberrant or even nominal activity drives disease. Historic advances in life science research have been made by application of biological techniques, but modern research relies heavily on interdisciplinary methods. These are computational things such as computational biology, biophysics, or something that has been coined chemical biology, which is what my lab applies. The exact definition is somewhat subjective, but I like to simply define it as the application of chemistry that does not naturally occur to learn more about living systems. The biology that occurs in cells is extremely complex, so to maintain a tractable research program, labs typically focus on a particular aspect. In my case, this is something known as post-translational modifications, or PTMs. The importance of PTMs can be put into context if you look at this diagram depicting the central dogma proposed by Francis Crick. Crick correctly stated that DNA consists of protein coding genes, and these are first copied to RNA, and then this message is used as an instruction manual for protein synthesis. However, there are not enough protein coding genes to support the complexity of higher organisms, and this discrepancy has been addressed by the discovery of further enzymatic chemical modification of proteins, and this is known as post-translational modification. The first PTM to be discovered is something known as protein phosphorylation. This involves the transfer of a small inorganic phosphate group to hydroxyamino acids and is carried out by enzymes known as kinases. Phosphorylation greatly expands the biological code of the cell and is involved in essential processes. Aberrant phosphorylation can drive diseases such as cancer, and a number of drugs in the clinic are small molecules that inhibit kinases. A PTM my lab studies is something known as ubiquitilation. Unlike phosphorylation, it targets lysine residues. Another distinction is that rather than the modifier being a small phosphate group, it is in fact a protein itself called ubiquitin. Hundreds of proteins are involved in ubiquitin attachment and removal, and we are interested in the enzymes that attach ubiquitin, known as efreeze. If you are wondering why we represent proteins in this ribbon-like representation, it's simply because visualizing them in terms of their atomic components is information overload in most cases and makes the overall fold of the protein, which is of primary importance, clearly visible. This is the scale and illustrates the difference in size between an inorganic phosphate group and the ubiquitin molecule. There's a huge difference, as you can see. Efreeze do not work in isolation and are dependent on an upstream E1 and E2 enzyme that juggle the ubiquitin between each other, and the ubiquitin is passed on like a baton to their respective active sites. When a substrate protein is post-translationally modified with ubiquitin by the E3, it is typically degraded and recycled in the cell. E3s are often upregulated in cancer and promote other diseases such as muscular and neurodegeneration, so are compelling drug targets. E3 hijacking is also a new therapeutic strategy, so these enzymes are of great medical importance. To discover new E3s and learn more about existing ones, we develop chemical biology tools. One such tool involves chemical engineering of the upstream E2 enzyme that is loaded with ubiquitin. This allows us to measure this specific activity in this conjugation process. We chemically modify the E2 ubiquitin conjugate by inserting a chemical trap that bonds to the active site of a functional E3 enzyme. We also chemically install a chemical hook that allows visualization or retrieval. This engineered protein conjugate can serve as a probe for studying cellular E3s. An experiment we carried out was basically a fishing expedition where we added our probe molecule to an extract from mammalian cells, where enzymes that would have E3 activity would be trapped by the probe. By virtue of the retrievable hook, we could enrich these proteins that were labeled by the probe, which would be candidate E3 ligases. Using an analytical technique known as mass spectrometry, we could identify all of these proteins, these candidate efreeze. Known efreeze were identified, providing a powerful technique for studying their activity in various disease models. Unexpectedly though, a protein called MICBP2 was strongly labeled by the probe, but this protein was not thought to be an E3. We were very intrigued by this because it suggested that we have a lot more to learn about E3s. Furthermore, total removal of this protein had been shown to have important functions in neurons. In particular, its removal protects axons against neurodegeneration in models of injury. Using a combination of techniques, we determined the molecular structures of MICBP2 and delineated its ubiquitin transfer mechanism. Surprisingly, it carries out its own ubiquitin relay act as it juggles the ubiquitin molecule between two active sites. Unlike transfer to lysine residues like canonical efreeze, it actually transfers ubiquitin to hydroxy amino acids like the kinases, which was very unexpected. This unique mechanism could be disrupted with small molecules. So this offered a compelling therapeutic strategy if this ubiquitin relay activity was actually central to this axon degeneration role that had been assigned to the whole protein. We tested if this relay mechanism was central to this degeneration and found that all of the neuronal characteristics were recapitulated by simply inactivating the relay mechanism and leaving the protein otherwise intact. These experiments validate blocking of MICBP2 relay activity as a therapeutic strategy for a range of neurodegenerative disorders, and we are actively pursuing this. I would like to finish here and acknowledge members from my lab who contributed to this story, and also collaborators and the fantastic support services here at the University of Dundee. Thank you for your attention.